Good evening, everyone. We'll be starting in a bit. You guys can hear me, right?
Yeah, sure. Good evening, sir.
Sorry, sir, for disturbing. I am having one doubt about exam.
The exam, yeah, go ahead.
So, for how much marks we will having the exam?
I think the last I saw it was 60 marks for the last final exam and 40 from the assignment questions.
For animal physiology?
For animal physiology, I think it was given on the website. I can check it for you. Just let me look at
Sorry.
it.
Uh, so I can't hear you properly. Can you just repeat?
Just wait a minute. I will grab my headphones. I just wanted to show.
Okay.
Should we? Yes. No, it is 75% of the exam score. So the exam will be out of 100, it will be brought down to 75%. And out of the 12 assignments that you have been filling in week after week, 8 of the best will be chosen, and that total will be brought out to 25%. And both of them will be added, and you will be scored for a full 100. And if you score more than 40, then you will get a certificate from NPTEL.
Okay, sir.
Yeah,
So,
yeah.
the second question which is arising in my mind, how many questions will be there in the 400 marks?
So that depends on the, it varies every examination. And since I have not opted for one myself in this course, do not have much of an idea. Maybe you can post this on the NPTEL forum, and maybe the other TAs might be able to help you who are designing the questions for the final exam themselves.
Okay, sir.
Okay, I will just go and grab my headphone. Just wait 1 second and then we can start. ব্লুটুথ অফ করো, ফোন কোথায় তোমার? আমার তো কানেক্ট হয়ে গেছে। Yeah, can you guys better hear me this time around?
So screen sharing is visible over there.
Okay. So we had another question here. Let me see. Aruna is asking, sir, can we get the previous year's papers? You mean the final paper? I do not think I have access to that. I have access only to the assignment questions. This is, I think, another question that you can drop right into the NPTEL forum rather. Is that fine? Yes, Yash.
Okay.
Okay. Yash, do you have any question or no? Then you can. So the questions will not be
10 questions.
for the final exam. I do not think the
On
questions will
each
be picked and
assignment.
chosen from the assignment questions that you have been doing. The final exam will probably have different questions as compared to the assignment questions that you have been doing. But the topics will be same and the questions will be similar. So if you have been doing the assignment questions and reading up on them, I think you will be able to tackle whatever there is. Probably not. Probably not. The questions will probably not be repeated, but the concepts might be the same. Like it might be the same. The answer might be the same. The questions might be a bit, going in a turnabout manner. Like the yeah. Okay. So let us begin today's session. So hello everyone. Welcome to the week 12 of Animal Physiology and a PMRF-NPTEL session. And this week we are going to deal with the endocrine, digestive, blood, kidney, as well as reproductive system. So yeah, we have another question. English has type of questions of final exam, only MCQ subjective. Most probably it will all be MCQ, but again another question that can be dropped in the NPTEL forum because I'm not involved in making the final and sign question paper for the NPTEL exam. I do not have the type of question it will be, but most of the time these are MCQ based questions. Similar will be very similar to what we have been answering for assignments. Okay, so I will briefly introduce myself. My name is Anubhav Chatterjee. I am a Prime Minister Research Fellow and PhD scholar in the Department of Biological Sciences and Bioengineering, IIT Kanpur. So as you would know that we are holding this PMRF-NPTEL sessions to clear doubts of the students that are joining them, as well as trying to answer the assignment question of the previous year's run of this particular NPTEL course, Animal Physiology. And by answering the questions, we'll try to get a better understanding of the overall concepts behind the questions and how to tackle them during the exam. So as you would know, all of these sessions are recorded and the recording is available on this particular playlist on YouTube. This link is also available on the NPTEL web page, and you can go ahead and watch them whenever you feel that if you feel you have missed something or would like to go through something again, or if you have not been able to join the session, and drop whatever questions you have in the YouTube comments or the NPTEL forum directly. So Aruna is asking, sir, in what way these certificates are useful? So most of the time, these are very useful in your CV, that you can show that we have actually covered the course from somewhere legitimate, and with the you know uh, with the academic credits, the online credits, the ABC, being held up nowadays to catch all of the credit score of students, how many courses that they have gone through. This will also help you to do that as well, that you can apply for NPTEL courses and put them into the ABC credit system as well, and that will also use for help for your CV. Okay, and it will also help you study for if you have to take a higher competitive exam, then you will probably have an edge from all the material that you have been learning. But most of the time, this will only help your CV and your general sense of the subject itself. Okay, is that okay? Okay, so does anyone have any questions for the previous week's lectures? Then we can tackle them and then move on to this week's assignment questions. Okay, I think no one has any questions on this week's assignment questions. So we'll go to the first one. So how do we do this is that I will be asking a question such as this. You will have four options A, B, C, D, and I will encourage everyone to put in whatever they think is the right answer A, B, C, D into the chat, and then we will discuss the question itself. So the first question is erythropoietin is a blank that is produced in blank. The options are erythropoietin are RBCs produced in the heart. Erythropoietin is a hormone produced in the kidneys. Are RBCs produced in the lungs, and erythropoietin is a hormone produced in the brain. So you can put in whatever you think is the correct option into the chat. Aruna says it is B. Erythropoietin is a hormone that is produced in the kidneys. Anyone else? Vishwajit also says it is B. Erythropoietin is a hormone that is produced in the kidneys. Does anyone else want to give it a try? Okay. So if we just look at erythropoietin itself and break the name down, then you will understand that erythro actually means red blood cells, and poietin means to make. So essentially, the entire word comes together to tell you that erythropoietin is something that helps to make RBCs or red blood cells. So major producers of erythropoietin is the kidney, and a little bit comes from the liver. And how this hormone acts is that it goes and acts upon the bone marrow. So bone marrow is the squishy part in the middle of your bones, and this is responsible for making new blood cells, WBCs, RBCs, or platelets. And under the influence of erythropoietin, this particular bone marrow cells, or BMSCs, start making more and more red blood cells. So all the cells here will start to differentiate into red blood cells, but initially these are immature red blood cells, and later on they mature into the red blood cells that we know and which carry oxygen and carbon dioxide. So how does it exactly work? So there are various sensors in our body which detect the amount capacity of oxygen, the saturation of oxygen, and the affinity of oxygen that is present in our blood. So essentially they monitor how much oxygen the blood is carrying and how much it can deliver. If the blood carries less oxygen than is required by the body, that is known as hypoxia. Hypoxia is the condition in which the body is oxygen starved. In this particular condition, the kidney is able to sense this via various pathways that we are not going to discuss, but essentially the kidney is able to sense this and will start producing and secreting the hormone erythropoietin. Erythropoietin that we have just discussed will go and act on the BFU, the bone marrow blood forming unit E, or erythropoietin. And this will ask these particular cells which are present in the bone marrow to start dividing into further and further differentiated forms of themselves, finally forming the reticulocytes. And these reticulocytes will divide and form our RBCs. So this is how the entire process works. The sensor in the blood detect there is a low capacity of oxygen. This message is transferred to the kidneys. Kidneys release erythropoietin. Erythropoietin goes and binds to the BMSs in the bone marrow. The BMSs differentiate to form uh, further and further differentiated forms of themselves, which ultimately form RBCs. So is that okay? Is that fine? Okay, I hope that is okay. Thus, if we go back to the question, erythropoietin is a hormone that is produced in the kidneys. Anyone has any questions from this? Okay. Moving on to the next question. Tamoxifen, a dopant banned in sports, is mainly a blank that is used commonly in the treatment of blank. The options are: tamoxifen is a pregnancy inducer used in treatment of ALS. It is an estrogen modulator used in the treatment of ALS. It is a pregnancy inducer used in the treatment of breast cancer. It is a estrogen modulator used in the treatment of breast cancer. Estrogen modulator and used in the treatment of breast cancer. So Jagruti says it is D. Tamoxifen is an estrogen modulator used in the treatment of breast cancer. Anyone else? Anyone else wants to try? Okay, so let's look at it. So tamoxifen, the molecule, is a synthetic analog of estrogen. A synthetic analog of a drug is one, or analog of a particular biological molecule is another synthetically made molecule or biologically made molecule that can take its place and act on the same pathway but sometimes have different effects. So you can consider them being a cricket ball compared to a tennis ball. Both you can play cricket with both, but both are different in nature. But most of the time they can be interchanged, but sometimes they have different actions. Okay, so estrogen, the normal function of estrogen is during breast cancer, estrogen is overproduced, and estrogen can bind to the estrogen receptor as shown right here, and the receptor dimerizes and goes into the nucleus, initiating and asking the cells to proliferate a lot. This is essentially one of the hallmarks of estrogen reactive breast cancer or ER. So these guys will proliferate because of overproduction of estrogen, and these continue to produce the estrogen themselves. So, mechanism of action of treatment of breast cancer is via the use of tamoxifen. So, tamoxifen, as I said, is an estrogen analog. So, estrogen instead of estrogen, tamoxifen can go and bind to the estrogen receptor. But the problem here is that during the dimerization, the tamoxifen bound receptor cannot recruit the necessary co-activators to initiate cell gene transcription and express the gene necessary for cell proliferation. So you essentially replace the estrogen with the tamoxifen, which went and bound to the receptor but cannot do any of the downstream activities. So the entire cell proliferation regime of the cancer cells is stopped. And this entire idea is used in the treatment of breast cancer cells which are estrogen positive or ER positive. Is that fine? Does anyone have any questions from this? Okay. So tamoxifen is a dopant. So because of the activity of tamoxifen as an estrogen modulator, it is used as a doping agent. So it increases the stamina and the endurance activity of the body in addition to the other factors or other characteristics that we have just discussed. So tamoxifen in that regard is used as a doping agent to increase the endurance and the stamina of the athletes that are competing in a competition. So tamoxifen is a banned dopant in sports and is mainly a modulator that is commonly used in the treatment of breast cancer. Any questions from this? Okay. So next, plank is used to increase milk production in cattle. The options are serotonin, oxytocin, GABA, or glutamate. You can put whatever you think is the correct option into the chat. Jagrut says it is B. Oxytocin is used to increase milk production in cattle. Anyone else? Anyone else wants to try and answer this? Charge it up. Anyone else wants to try and answer this? Okay, so the correct option is oxytocin, as Jagruti has already said, and it is a peptide hormone released from the posterior pituitary gland, which is present at the base of our brain. So here is the posterior pituitary gland, as you can see right here, and this particular hormone is secreted during pregnancy, and it helps in two different ways. First, it helps in uterine muscle contraction to actually initiate the pregnancy, which actually helps in pushing of the baby out of the mother, and it also helps in the production of milk. So it has dual functions, but essentially it is very, very important for the final stages of pregnancy as well as milk production. And this particular action of oxytocin has been used uh, a lot in order to produce extra milk in cattle, because in cattle which are not pregnant, even in them, if oxytocin is given, the milk production increases because the body thinks that the cattle is going to have a calf, and that is why it produces a lot of oxytocin, a lot of milk in response to the oxytocin. So the correct option is B, which is oxytocin. Any questions from this? Okay, I think there are no more questions, so we'll go forward. Blank is a chain of 2 to 50 amino acids. The options are A, aggregate, B, agar, C, peptide, D, proteome. Aruna says it is C, peptide. Anyone else? Jagruti also says it is peptide. Kamlesh also says it is a peptide. Okay. Anyone else want to try and answer this? Okay. So let us look at it. So we had covered the structures of various proteins in the way back in the first PMRF-NPTEL session that we did, and in that we learned that structure of protein exists in four different stages. So there is a primary structure, there is a secondary structure, there is a tertiary structure, there is a quaternary structure. So the primary structure is made out. So the protein is essentially a polymer of different individual amino acids. So these are these discs, colored discs here represent the different amino acids. Our genome codes for exactly 20 of the amino acids, and these are joined together to form proteins. But they are joined together with something known as a peptide bond between them, where the carboxylic acid group interacts with the amine group to form the peptide linkage, and they form these long chains of amino acids, as we can see right here. This is if this entire structure is small, 2 to 50 amino acids is known as a peptide, and this is the primary structure of a amino acid, of a protein. Next, we have a secondary structure in which this there is localized folding in this peptide chain. So it folds to actually envelop the hydrophobic ends inside and the hydrophilic ends outside of the protein, and that actually forms these localized clusters. So you probably would have heard the beta sheets, the alpha folds, all of these that form as part of the protein secondary structure. Next, what happens is that the secondary structures all across the protein. So this a protein is huge. If you have to think about it, it ranges in the kilos of Daltons. So there are almost thousands of these peptides joined together. So essentially, the first stage is joining of the amino acids to form small peptides. The peptides join together to form a large peptide. The smaller parts of each of the peptides all uh, the smaller parts of each of the peptides fold to form the secondary structures. The secondary structures interact to form the tertiary structures. Different tertiary structures interact to form the quaternary structures. So there is a step-by-step fashion in which the protein folds, and the tertiary and quaternary structures are the only ones that are functional. All the previous structures are not functional. So if you go back to our question, blank is a chain of 2 to 50 amino acids, and the answer is peptide. So here we know that agar is a protein that is agar is a kind of polymer that is taken out of seaweeds, which helps in uh, I'm missing the word here, which helps in gelling together various electrophoresis gels that we use for running DNA as well as proteins. Peptide is a 2 to 50 long chain of amino acids. Proteome. So, can anyone tell me what a proteome is? Uh, does anyone have an idea what a proteome is? Okay, so Kamlesh says agar as a nutrient medium. Okay, so agar by itself, so if you would have heard of agar as a nutrient medium, you probably are referring to LB agar or Luria bertoni agar, which is used to make bacterial plates in which we grow bacteria. So in that particular media itself, the actual nutrients come from the LB broth. The LB broth contains peptides, yeast extracts, tryptone, all of these things from which the bacteria actually take nutrients. The agar itself is added to just solidify the entire thing into plates. So if you have ever seen agarose plates, those are jelly-like structures. But the LB broth is liquid. To make them into a jelly-like structure, solidify them, we use agar. Agar by itself does not act as a very good nutrient medium. Is that okay, Kamlesh?
Yes, sir.
Okay. And the last part is a aggregate. So aggregates are sorry, someone that wants to join. So okay, yeah. So the last of them here is aggregate. An aggregate is a type of a misfolding of a protein. So say a protein folded into its so out of the so as we had just discussed, the proteins can first form a primary structure, form a secondary structure, then a tertiary, then a quaternary. But upwards of the secondary structure formation, you can understand that there are multiple ways in which the protein might interact and fold. So folding is essentially you try to get the hydrophobic ends inside and the hydrophilic ends outside. So most of the time this process goes as intended and you have functional proteins. But sometimes what happens is that proteins aggregate. The hydrophobic ends are inside and the hydrophilic ends are outside, but in a fashion that is not functional. And these forms insoluble aggregates of proteins. So these are clumps of proteins that form throughout the cell, and these mm, are not functional and essentially disrupt a lot of cellular processes. And this is one of the reasons behind the diseases such as Parkinson's or Alzheimer's, where protein aggregates of the tau protein occur and accumulate throughout the cells, and these cannot be cleared out by the cells by themselves, and these interrupt with different uh, microtubule transport system of the cells and overall make a nuisance of themselves, and that essentially finally causes the cell to die off. So that is what an aggregate is. But for our particular question, blank is a chain of 2 to 50 amino acids. The correct option is peptide. Anyone has any questions from this? Okay. Moving on, renin and angiotensin are produced by the options are pituitary gland, kidneys, pancreas, and ovaries. Aruna says it is produced by the kidneys. Anyone else? Vishwajit also says it is B, kidneys. Kamlesh also says it is B, kidneys. Renin and angiotensin are produced by the kidneys. Jagriti also agrees that it is B, the kidneys. Okay, so let us just look into it. So renin and angiotensin system is essentially a feedback loop which essentially modulates the amount of sodium as well as water deabsorption by the kidneys. So, just a basic understanding, what is the reabsorption that the kidneys do? So, okay, that doesn't sound like lunatic ideas. If someone is doing something, please let me know. Okay, I will get back to you, Dilly. I will get back to you just a bit. So, kidneys essentially function as a huge filter for the entire blood in our body. So it filters out essentially the function of the kidneys is to maintain a particular composition of the blood, and it tries its best to maintain the specific balance of various salts as well as water as well as cells in the blood, which is known as osmoregulation. So how the kidneys do it is that they have two different steps. You can separate them as two different steps. In the first step, the kidney filters out a lot of water as well as a lot of salts. So you have sodium, potassium, calcium, as well as a lot of water flowing through the kidneys into something known as the glomerulus. This is the first filtration step. The glomerulus then goes via the loop of Henle into the bladder. But while in this passage, the kidneys will continue reabsorption back of some of the water and the nutrients as well as different salts present in this filtrate. It is done so that it is done very specifically what will be reabsorbed by the kidneys, and it is done so that you can maintain a very good balance or maintain the osmoregulation of the blood. So if more of the water has been filtered out, it will reabsorb a lot of water from the loop of Henle. A lot of sodium and different proteins have been filtered out. They will be reabsorbed by the loop of Henle and the other structures in the nephrons. So this is what is reabsorption via the kidneys. So how does this work? So if there is a drop in blood pressure or a fluid volume, so there is problems in the osmoregulation, the kidneys release the protein known as renin. So renin goes and acts on angiotensinogen. They are released by the liver. So a lot of names you do not have to bang your head on it. Essentially, once there is a problem in osmoregulation, renin is released, which acts on angiotensinogen, which converts into angiotensin 1, and angiotensin 1 is converted into angiotensin 2 by another enzyme. Angiotensin 2, it goes and causes something known as vasoconstriction. It constricts the blood vessels, which allows more and more water to be. This increases essentially the blood pressure, and along with angiotensin 2 acts on the adrenal gland sitting on top of the kidneys to release the hormone aldosterone. Both of these actions come together to increase the reabsorption of sodium and water. Essentially, kidneys realize that there is a lot of water and sodium that is going out through the filtration system. It doesn't want that much water and sodium to go out, so it releases renin, which through a cascade forms angiotensin 2, which acts on the kidneys to allow for reabsorption of the water and the sodium that was lost. So the renin-angiotensin system essentially helps in maintenance of the osmoregulation via reabsorption of water and sodium. Any questions from this? Anything that you did not understand? Does anyone want to ask anything from this? Okay. Renin and angiotensin are produced by thus the kidneys. Moving on, we move on to the thyroid uh, gland and its hormones. The thyroid releasing hormone TRH, the thyroid stimulating hormone TSH, and T4 work together to. So the thyroid releasing hormone from blank, thyroid stimulating hormone from blank, and T4 from blank work together to maintain homeostasis. So which of the glands are we talking about here? So blank here, blank here, blank here. TRH is secreted by what? TSH is secreted by what? T4 is secreted by what? So the options are hypothalamus, pituitary, thyroid, and jumbling up of the three. So Aruna says it is A. Hypothalamus releases TRH, pituitary releases TSH, and thyroid releases the T4 as a result of both of these. Kamlesh says also it is A. The hypothalamus TRH, pituitary TSH, and thyroid T4. Jagriti also agrees with the answer that is A. Anyone else? Okay. So this is essentially the entire cascade. So the hypothalamus acts as the initiator in this cascade and releases the thyroid releasing hormone, which acts on the anterior pituitary. So anterior pituitary is the master gland in the body, and the hormone released by the anterior pituitary acts on other glands and asks them to release their hormones. So anterior pituitary releases the hormone TSH, or thyroid stimulating hormone. This thyroid stimulating hormone goes and binds to the thyroid gland present in your neck, and thyroid produces T4 and T3, which are two variants of thyroxine, and T3 is the active version, and so T4 and T3 both are released. Once reaches its site of action, it is converted to T3 and can act on various cells in the body. And all of this is connected by a feedback loop. A lot of T3 or a lot of T4 can have inhibitory effects on anterior pituitary and the hypothalamus, telling them that there is a lot of thyroxine in the blood already, we do not need any more. So thyroid TRH is stopped, which in turn can stop the TSH, and thus more thyroxine is not released. So the entire thing goes via a negative feedback loop. So this is how it works: TRH by the hypothalamus, TSH by the anterior pituitary, T4 and T3 via the thyroid gland. Any questions from this? Okay, I think it's all okay. So thyrotropin releasing hormone TRH from the hypothalamus, thyroid stimulating hormone from the pituitary, and T4 from the thyroid gland work together to maintain homeostasis via negative feedback loop. Moving on, iodine deficiency leads to blank that primarily affects the blank. Iodine deficiency leads to goiter, primarily affecting adrenal glands, scurvy, adrenal, goiter, and thyroid, scurvy, and thyroid. So Jagrati says it is C, iodine deficiency leads to goiter that primarily affects the thyroid gland. Kamlesh and Aruna also say the same, that iodine deficiency leads to goiter and primarily affects the thyroid gland. Anyone has any questions? Any other options that they want to put into the chat? একটু আসতে Okay. So if there are no more options that anyone wants to give in, so let us look at it. So goiter is the condition in which there is a deficiency of iodine in your diet. So this essentially feeds as a tightness in your throat. So there is a swelling in your throat, and this can increase to even a protrusion in your throat that is quite big, and it causes difficulty in swallowing. It causes difficulty in coughing, hoarseness, and difficulty breathing. And this has other uh, effects such as un like very fast weight gain, tiredness, lethargy in doing anything, and many more other symptoms. And goiter can look as an enlarged thyroid gland. So your thyroid gland increases in size, which actually causes the swelling, and which is result in a visible swelling at the back of your neck that is obvious from far away. So this is what a healthy thyroid looks like, and this is what a goiter thyroid looks like. It essentially swells up. So what is the reason behind thyroid? It is the deficiency of iron in your diet. Where does that come in? So if you look closely into the thyroxine molecule, you see these groups right here, I. So essentially, thyroxine requires a lot of iodine to make. So without iodine, you do not have any thyroxine that is getting made. And this is what essentially causes goiter. Low amounts of iodine causes deficiency in thyroxine, and this causes all of your problems from goiter. So, yeah, iodine deficiency, you have goiter because there is no iodine to make more thyroxine. The entire, yeah. So the thyroid glands essentially enlarge because they want, or there is a dysregulation in the entire process. And so, as we have seen in this particular diagram, that TSH acts on thyroid to form T4 and T3, which cause an inhibitory feedback loop. Once T4 and T3 are not getting made, you actually do not have any inhibitory feedback loop. The body thinks that the thyroid is not doing its job and continues to pump TRH and TSH into the blood, which go and keep on acting on thyroid. So how TSH works is that it causes the cells that actually produce thyroxine to grow bigger, proliferate more, and produce more thyroxine. But without any iodine, you are not actually producing any thyroxine. So TSH actually causes these cells to proliferate more, get bigger in size, but not release any thyroxine. And this particular phenomenon continues to develop more and more till thyroid gland itself is huge because of these thyroxine producing cells becoming more in number as well as size. But this entire thing is not inhibited because there is no T3 or T4 to stop the hypothalamus or the anterior pituitary to pump more of these hormones into the blood. Is that fine? Or should I go? Yeah, should I explain it again or is it fine? Okay. So iodine deficiency leads to blank that primarily affects the plant. The iodine deficiency leads to goiter, which affects the thyroid glands. Okay, moving on. The parietal cells secrete blank, and the gastric chief cells secrete blank that is further converted into blank. The options are parietal cells secrete HCl, gastric cells, chief cells secrete pepsin, which is converted to pepsinogen. And we have jumbling up of all of these. So Aruna says it is D. Parietal cells secrete HCl and gastric chief cells secrete pepsinogen, which is further converted into pepsin. Kamlesh and Jagruti also say the same, that parietal cells release HCl, gastric chief cells secrete pepsinogen, that is further converted into pepsin. Anyone else wants to try and answer this? Okay, so parietal cells and gastric chief cells. So both of these cells are present in the lower part of your stomach in essentially a groove-like structure which forms ridges at the end of your stomach. And firstly, we'll talk about the parietal cells. The parietal cells are the ones that essentially produce all the HCl which is used by the stomach to digest your food. So as you would know that the stomach produces a lot of HCl which digests all of our food, and it is very very essential for the entire process. And these are the cells behind production of HCl. So HCl, by you would know that it's a very strong acid, and producing and storing it is very difficult because the cells would probably die from the harm that the HCl is producing, that the produced HCl will cause on them. So the HCl produced in situ, that is when, as and when required, right at the surface of the stomach. How that occurs is that the parietal cells line the inner lining of the entire stomach, and actually there, this side is the stomach, the left hand side is the stomach, the right hand side is the interstitial space or towards the interior of the body. So essentially what they do is that they have two different types of pumps. The first is a chloride pump that pumps chloride ions into the lumen, as well as potassium and hydrogen pumps. So we have looked at these kinds of pumps before. So these are ATPase pumps which allow the movement of ions against their concentration gradients. So if you remember, potassium is higher inside the cell and lower outside the cell. So using pumps such as this and usage of ATP to you know cause the pumps to work, more and more of potassium is pumped inside the cell, while hydrogen ions or protons are pumped out of the cell. And this continues till you have a lot of accumulation of hydrogen plus ions and chloride minus ions, and both of these combine in the lumen of the stomach to form HCl. And there is a lot of other functions going on this side, which essentially boils down to maintaining the homeostasis of the cell to make sure you do not have a lot of potassium ions so that it can keep on doing this function and produce a lot of like takes in a lot of chloride ions so that it can pump it towards the lumen of the stomach. So this is how the parietal cells function. Any questions from this? Okay, then no question. We'll talk about the next type of cells, which is the gastric chef cells. So the gastric chef cells look something like this, which they have a very polarized look. what a polarized look means is that one end looks very different from the other. So you can see one end has ridges in them, which increases the surface area, and the other end is just does not have any ridges, and this attaches to the basement membrane at the end, uh, at the stomach lining. So, and these cells, so as I said, your stomach is lined with the grooves such as this. As you can see right here, there are grooves that go deep into the stomach lining. This is all done to increase the surface area of the cells that are exposed to the stomach lumen. And most of the gastric chief cells are located in the interiormost part of the ridges. Here, 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 here. Essentially, the action of the gastric chief cells is to produce pepsinogen. Pepsinogen is a pre enzyme which, in the presence of HCl in the stomach acidic conditions, it is converted to pepsin. And pepsin, as you would know, is a enzyme which can chew up proteins. So it chews up big proteins in our stomach and produces short peptides out of them. So when we ingest our food, it is made out of essentially a lot of proteins. And these proteins are digested majorly via the pepsin released in the stomach, and these produces short peptides. Once these short peptides are produced, this is further broken down in the small intestine via various other enzymes such as carboxypeptidase. So different peptidases are there which break these short peptides to the final amino acids that we know, and these are reabsorbed in the small intestine. So the parietal cells secrete HCl, gastric cheese cells secrete pepsinogen that is further converted into pepsin. So the right answer is D, as everyone had said. Any questions from this? Okay, there are no more questions. Moving forward. Blank emulsifies fat for digestion, and blank that controls blood sugar. So what emulsifies fat for digestion and what controls blood sugar? The options are bile and bilirubin, bilirubin and insulin, erythropoietin and insulin, and bile and insulin. Aruna says it is B, bilirubin and insulin, and Jagruti also says it is B, bilirubin and insulin. Aruna changes her answer to D, bile and insulin, and Kamlesh also says it is bile and insulin. Anyone else want to try and answer this? Anyone else want to try and answer this? No? Okay. So, yeah, we'll first talk about the bile. So, bile is a particular secretion that is produced in the liver, which follows the breakdown of RBCs. So, all the RBCs that are broken down, they are broken down, the cellular component is extracted from the hemoglobin. The hemoglobin is then broken down into its constituents, the iron Fe2 plus and the heme group. The heme group is converted to bilirubin and finally to bile. And this bile is produced by the liver and stored in the gallbladder. And this, when digestion is underway, the gallbladder releases the entire bile back into the stomach as well as the small intestine. And this entire thing, what it does is that it helps with the digestion, and it does something known as emulsification. So our body, the food we digest, that also contains fats and lipids. So the HCl is not very effective in on acting on fats and lipids, mostly because fats and lipids are not water soluble. So if you want to digest or metabolize or you know absorb the fats and lipids, you need them to be in a water soluble form. So what the this bile does is that bile has two separate different groups at two ends, a hydrophilic group and a hydrophobic group. The hydrophobic group binds to the fats, and the hydrophilic group forms a membrane around the fats. So you essentially have globules or uh, droplets of fat that are present in the digestive tract. So you probably would have seen, if you have ever mixed oil and water and shaken them very hard, then you see small bubbles made out of oil. Those are the micelles that we are talking about. But these ones are much more stable and are formed in the lower intestine and absorbed in that form, in the micellar form, via the lower intestine. So essentially, bile helps in the absorption of fat via emulsification and solubilization of the fat. And finally, it also helps in excretion. Major composition of bile are bile salts, various salts that are produced by digestion of RBCs as well as end products of metabolism, phospholipids, cholesterol, and bile pigments, bilirubin. So bilirubin is what gives it the color, which is a yellow color after the metabolization of the heme group of hemoglobin. It forms a yellow colored pigment, which is known as bilirubin, which is present in bile, which gives it the yellow color. So going back, okay. So the other question was what helps in glucose absorption. So the glucose absorption enzyme is known as insulin, as we would all know. So what happens is that once we take in food, our blood glucose rises because all the lower intestines, everything are absorbing a lot of nutrients and glucose back into our blood, and these are all released into the blood. But how do exactly the cells start absorbing the glucose to initiate their metabolism? That is done via insulin. Insulin goes and binds to various receptors on the cell surfaces, I see right here, which asks the cells to start taking in the glucose and using the glucose. So once you release insulin to the blood, the and essentially the glucose receptors or the glucose transporters on the surface of the cell open, and glucose enters into the cell and is metabolized. The opposite function is done by glucagon. So glucagon acts when there is very less amount of blood glucose, which is in the middle of in between two meals. So there's less blood glucose because there's no digestion going on, and glucagon does the opposite. It asks the liver and various cells to release glucose. Glucose goes into the bloodstream, maintains an equal amount of blood glucose. And obviously, it also talks, we can think about is how exactly the related disease works, which is diabetes. So, can anyone tell me what are the two types of diabetes that we have? Types of diabetes. Does anyone have any idea? T2DM, type 2 diabetes, mellitus. Yeah, mellitus is one of them. And T1 is insipidus. You are absolutely correct, Aruna and Kamlesh. So T1 will be not DM but DI. Kamlesh. So type 1 is diabetes insipidus and type 2 is diabetes mellitus. What is the difference between them? Both of them are because insulin is somehow non-functional on the body. So there's a lot of glucose that is going around in the blood of our body without being reabsorbed into the cells because somehow insulin is not working. In type 1, in type 1 diabetes, our cells, the immune cells of our body, for some reason or other, start attacking the beta cells of the islets of Langerhans. These are the cells that produce insulin. So once the immune system start attacking your own pancreas, no more insulin is produced, and that causes no reabsorption of glucose or absorption of glucose into cells. So you have a lot more blood sugar or blood glucose going around in your blood, and which causes type 1 diabetes. The type 2 diabetes, which so type 1 is majorly genetic or and majorly occurs from a younger age when such changes takes place. Type 2 is something which is very commonly linked to the Indian population and very commonly linked to the metabolism and lifestyle choices of a person. In the type 2 diabetes, what happens is that due to two reasons it happens is first, there is less production of insulin by the beta cells of the pancreas. This might be genetic or due to accidents or due to environmental causes. But the other reason is much more common, in which the body does not recognize or resists the amount of insulin being produced by the beta cells of the pancreas. Majorly, it is because of lifestyle choices which causes obesity in older, like in mature humans. So due to lifelong obesity and inflammation throughout the body of adult humans, what happens is that there is a lot of insulin that needs to be produced by the beta cells of the pancreas. Lot of insulin being produced causes insulin resistance. The cells do not recognize insulin anymore because they are already accustomed to a lot of insulin, and that causes the second type of diabetes, which is diabetes mellitus, and that again causes cells not to absorb glucose into themselves and increases the blood sugar in blood sugar. So two types of diabetes. Second one, we are sitting at the hot spot of diabetes. It is said that in the next 10-20 years we will be the highest. We are, I think, already the highest amount of we have the highest amount of diabetes patients throughout the world, and we are prone to become a hotspot for diabetes in the next 20-30 years if no one fixes the problem. So yeah, any questions from this? Okay, someone wants to join. Let me know. uh, yeah, in between, if anyone has any questions, then they can ask. Okay, if there are no questions here, then we can move on to the next one. Hmm. So blank emulsifies fat for digestion and blank controls blood sugar. The answer is bile emulsifies fat for digestion and insulin controls blood sugar. Any questions from this?
No, sir.
Okay. Final question for the day. Is it recommended to it is recommended to avoid aspirin in case of injury because causing cuts, because the options are: it is an antibiotic and causes infection, it is an anticoagulant and will lead to blood loss, it has an effect on heart and decreases blood pressure, it will cause atherosclerosis. Aruna says it is C, it has an effect on the heart and will decrease blood pressure. Kamlesh says it is B, it is an anticoagulant and will lead to blood loss at the site of injury. Anyone else wants to try to answer this? Okay, so the correct answer is the aspirin is an anticoagulant that will lead to blood loss at the site of injury. That is what the function of aspirin is, other than as a pain reliever. It actually involves a lot of how exactly aspirin works, and that actually causes it to also act as an anticoagulant as one of the side effects. Okay, thank you everyone for joining for today. Does anyone have any questions from this slides? Okay, so I would just like to ask you, when is the exam exactly? Do you guys have any idea? The final exam for NPTEL? 26th. Okay, so next Tuesday, we are going to have a final PMRF-NPTEL session in which I will go through the key concepts of the all the weeks that we have already covered, the 12 weeks, and we'll have an extra session on that. And I hope that I encourage everyone to try and join. I will be talking about everything that you need to know, and it might help a lot for the final exam that you're going to have. Okay, everyone, thank you for joining, and I will see you again next week. Thank you.